Hi folks, this is Beverly. This week we are going to talk about genetic technologies and testing. I just would like to tell you that I hope the first 2 weeks have gone well and you all are having some good discussions in your discussion group this week with the um, video that you're watching from the BBC. This is week 3 and week 3 starts on Monday. This week you will have a quiz. It will open on Wednesday and you'll have till Sunday at 11:30 p.m. to complete the quiz. If you read your book, listen to the lecture, you should be able to do quite well on the quiz. All the questions are taken from the text, um, but I am not going to cover all of the text and all of the possible questions for the 10-point quiz in this lecture. Um, so I know this week it's the only week you have 2 chapters to read, and we will not have a discussion in your discussion groups this week because of that. So here we go. So basically, first of all, we're just going to talk about DNA. We just need to understand that DNA is our instruction manual for our existence here in the world. It's what organizes and manages our bodies, and it's what keeps us alive. Um, all life uses some form of a genetic code, a DNA, for example. And your book talks about yeast in the fermentation process of making wine. We need to understand some technologies. There's biotechnology for definition, the use or the changing of cells, and biological molecules for a specific need. There is genetic engineering. This is a specific form of biotechnology that manipulates the DNA. There's transgenic, which is where we go from one organism that has DNA to another species. And recombinant DNA is artificial DNA, which is man-made and created. So we need to talk a little bit about, with all this new technology and all this new findings, that um, there are patents that have occurred in the field of genetics. With new recombinant DNAs being formed, they're considered to be intellectual property of the research facility or the particular individual researcher, um, and therefore it can have a patent. If you look in your book, there's a timeline that's provided of some specific patents that have come about or some specific laws that have occurred in relationships to patents and genetics. We do need to just keep in mind one interesting thing is that in 2009, patent law was challenged by the ACLU and the Public Patent Foundation after the discovery of the BRCA1 and BRCA2 genes, because the company Myrid Genetics and the University of Utah had the genetic patent on that particular genetic test. Um, and so it prevented other companies from being able to do that type of genetic testing, and the consumer itself could only receive that testing from Myrid. And the cost could reach $3,000. So the ACLU and the PPF felt that it was unfair to individuals who may carry the gene and might prevent some people from getting tests, and the costs were prohibitive, and so there needed to be a second genetic option. Additionally, with the patents out there, there was the development of what's termed the patent thicket, which means there may be multiple patents on the same gene. And this can happen because one particular person may have the patent on a particular gene sequence in which a researcher wants to look at a specific gene in that. And there has to be permissions given. There's often a replication of processes and some delay in research having to weed their way through the thicket of the patents. So we'll just talk briefly about amplifying DNA. The first known way to amplify your DNA is the polymerase chain reaction, and these take place outside of the cell. It uses the basic process of DNA replication, only it's able to do it at a much more accelerated rate, and it replicates a specific sequence of the DNA. It uses heat to keep the DNA from breaking apart during this reaction. I want you to understand you don't need to know, and memorize the steps of a polymerase chain reaction. Just familiarize yourself with the concept and look at table 19.1 for some uses of polymerase chain reactions. This is a schematic, a visual of what amplifying DNA looks like. We can also modify DNA. Um, we can do that by adding one genome to another. This was first done by using bacteria to produce peptides and proteins, such as what are used in medications such as insulin. Um, so then there's recombinant DNA technology, and this needs a restrictive enzyme, and it will cut a donor and recipient DNA in the same spots. And they work as scissors, and then they'll create these sticky ends, and then some cloning vectors, which are natural tools and carriers from one DNA from one species to another. And then we have common vectors and a plasmid, which is a small circular double-stranded DNA, and this exists naturally um, in nature. So we use recombinant DNA because perhaps we want to clone, we want to make specific copies of a DNA sequence. We're going to make specific gene products such as insulin, or we're looking to modify just a specific gene of interest that we want to research. Um, and that's why we would modify DNA. So we can monitor DNA function, and this looks at how we look at how genes express themselves. When we look at gene expression, we use the messenger RNA, and we use a term called microarrays, which create a profile of the expression of the gene in a small piece of the DNA. Um, so this can tell us how certain specific areas within the DNA sequence express themselves. And in looking at your textbook and how it applies to medicine, is that they're using this type of monitoring gene function for spinal cord injury. So they look at certain areas of gene expression to see how soon um, and where the healing might begin after a spinal cord injury, with the hopes that if we can modify the gene expression to limit the damage and increase the healing either more rapidly or sooner, perhaps we can prevent some of the complications of spinal cord injuries. The other thing we can do is the concept of silencing a DNA. And this is done, we want to diminish the expression or stop the expression altogether. And this is done on specific genes themselves. There are several ways to accomplish this. One way is RNA interference. And this is because the RNA is a short, double-stranded area, and it contains base pairs that are complementary. Um, we can create an interference with this, and it will halt the activity of the messenger RNA. We can also modify or stop the gene expression altogether, and this uses base pairs to block transmission or translocation of genetic material. So we'll look at nursing care related to people receiving gene therapy. So first thing we'll talk about is ethical considerations. We're going to spend some time in a couple weeks just talking about ethics in general related to genetics. And I'm sure we'll have some pretty hearty discussions. So a couple terms you've heard before is beneficence. Um, so we want to know if the benefits outweigh the burdens or the risks. And also we look at, is there a benefit to the future? So if we use this experiment on humans, what is the benefit to what we will learn in order to create an even better outcome in the future? And we have to look at that when we're using gene therapy or genetic modifications. We need to be sure that we honor the person's own rights to autonomy, making their own decisions, speaking for themselves, and we don't ever pressure a subject into either participating or not participating in any type of gene therapy or genetic research. We also look at justice, the cost of the research in the bigger picture of justice. Some societies would rather see research money spent for other causes rather than research in something that may not pan out. Recently in America, stem cell research gets a lot of, has had some of their funding curtailed because the public feels perhaps there are other uses for our financial healthcare resources. The other thing we look at in terms of ethical considerations and is informed consent. Um, we want to make sure that the patient is a willing subject. So we look at whether they're suffering, whether they're dying, whether we're going to give this person hope. Um, and we also, in this, talk about informed consent. Is it going to make their life or death more meaningful by adding to medical knowledge? Um, so we have to talk about that and um, make that part of the informed consent. We also need to be sure that we meet the obligations. There's a legal obligation of informed consent. Informed consent means that risks and benefits have been discussed. Additionally, alternative treatments have been discussed, and that in the process of informed consent, um, the promotion of personal choice has been foremost up front in the conversations. The patient also has to be of legal age and sound mind. One area of sound mind means that the person, if they're depressed, perhaps depression needs to be ruled out or that it's in the process of being dealt with. That means perhaps they're um, having some kind of support therapy, support group, or they might even be taking some type of antidepressive medication at that. So there is a protection for human subjects. There's a committee that decides if the expected benefits is worth the risk of the experiment on humans, and the committee must approve the research before it can even proceed, even if you have willing and informed participants. So some nursing care issues. If you're a nurse who happens to want to go into research or works with research, your obligation is to understand your research protocols and that you implement them exactly how they're intended to be implemented. Um, that the therapeutic results are adequately and thoroughly assessed and documented, that you document all side effects or unwanted effects and that they're monitored and managed. You also do complete and accurate documentation. And foremost, we provide psychological support for the patient as well as the family. So what are some standard gene therapies you might see out there that you don't even think about? Well, there are medications that can replace enzymes or hormones that the body isn't making for itself. For example, insulin in the use in the treatment of diabetes mellitus type 1. We can use recombinant genetic technologies, which you have bacteria that make the human insulin, and these bacteria can also make human growth hormone, which can be used to treat dwarfism. So, thinking ethically, um, should we use human growth hormone to treat shortness? Is it something that should be treated? It's always up for debate. There's never right or wrong answer. We can restrict intake of substances that the body cannot metabolize, such as in PKU. Although it may be difficult for the patient to follow restrictive diet and the medication regimens that it needs for it to live, there can also be medications that divert a chemical which is becoming toxic so that the body can, so it can be excreted, such as lactulose or kxalate. We have medications that inhibit enzymes that are harmful, such as lovastatin in relationship to cholesterol. We use direct removal of compounds from the body, such as when we use therapeutic plasmapheresis to treat familial hypercholesteremia, which when we move into our cardiovascular heart disease week, we'll talk extensively about. So then we ask ourselves, are we in the management of these issues or are we curing these issues? Medications are never replacement for what our own body or nature's own issue. So when patients have these diseases, they're managed, but they're never cured. They always are diabetic. They always have PKU. For example, in PKU, you may not get the severe retardation that would occur because of the lack of the ability to metabolize this an amino acid. But the children may still go on to develop some subtle learning disorders or may continue to have behavioral problems. Are there long-term consequences? Well, some treatments of genetic disorders can allow the manifestation of the disorder in another organ, or the treatment may lead to a different pathology, such as cancer. So there are some new treatments out there. We can maybe replace the mutant protein, such as with patients with emphysema who lack alpha-1 antitrypsin enzyme. We give factor VIII, transfusions to hemophilia A. Um, we can clone genes that are used to make, we clone the genes to make these relevant proteins. So are there any minimum requirements we need to think about when we're looking at the development of gene therapy? Well, it must be able to clone, we have to be able to clone the gene, and it has to be small enough to fit into the viral vector because the viral vector is what's going to carry it into the cell. We have to look whether or not this cure or management cost, risk, and benefit is better than the alternative ones that are out there. So we look at the risk versus benefit. We have to know, or the researcher has to know, the gene location. And there are multiple other requirements and concerns, as well as we need to have sufficient knowledge of the molecular basis of the genetic disorder itself, and if the gene transfer will correct the biochemical pathology. So is what we want it to do going to happen? There's sufficient regulatory components for the transfer of genes. How is it going to promote or suppress if necessary? Do we know the appropriate target cells to place the genes in? How long is the half-life? Do they have good replication potential? And we look at how that looks in vivo first. We look at the data of the cultured cell. We look at animal studies that the vector and the gene construct and the target cells, and how effective is it? Is it safe? After that, the protocols for human experimentation or human usage is approved by an institutional review board. And through the whole process, there's oversight by the government. So what is the main goal of gene therapy? To correct mutant phenotypes. So we can deliver normal genes to the somatic cells. And maybe, if we're lucky, we can correct it enough that we won't see the mutation in future generations. Are there any challenges? We don't know the number of cells that have to be corrected to get a good response. We need to compensate for the mutant gene because there's a loss of function even with the mutation. We need to be able to replace or inactivate, we could replace or inactivate a dominant mutant gene, and this could be even harder. We need to find the target cells that we're going to put the new genes into. We need to ensure a safe mechanism of transfer. So now we'll talk briefly about vectors. Vectors are the most effective means of gene transfer. They're used to modify specific cell types or tissues. They need to be manipulated. They are manipulated, and this will help change the expression of the therapeutic gene, the gene that we want to create the healthy response we want. And they're capable of entering every cell in the target population. And they've been engineered and they're able to engineered to render them incapable of replication. So they're able to keep the bad gene from replicating. There are some types of vectors. There's retrovirus, endovirus, and adeno-associated viruses. So what are the merits of viral vectors? They're non-toxic to the cell. On, they only a low number of copies of the viral DNA with the transfer gene are used to integrate into the host genome. They integrate well into their integrated DNA is very stable. So the retroviral vectors can accommodate rather large segments of added DNA. So we add the DNA to the retroviral vector, and this is large enough for many genes. Most retroviruses can't be used in non-dividing cells, which places a limitation on them. Although research is looking at lentoviruses to be in to go into non-dividing cells. There are adeno-associated viral vectors. They're a little bit safer. They do go into dividing and non-dividing cells. They're stable, but they hold a small amount of the DNA. There's adenoviral vectors. We can get these in high volumes. They will infect a wide variety of cell types, both dividing and non-dividing, and they can take an even larger, a 30 to 35 gigabyte of DNA. They can last a matter of weeks inside the target cell, and they're capable of causing a strong immune and inflammatory response to help with the process of the gene therapy. There are also non-viral vectors, which are naked DNA packaged in plasmids, liposomes, protein peptides, or on artificial chromosomes. Then this has been attempted. The DNA is generally taken up by the lysosome and then degraded. The DNA that remains in the proper tissue was not taken up effectively by the nucleus. The main use is for the treatment of cancer. So we're going to put this non-viral vector use a non-viral vector to get it into the cancer cell, and then it will actually kill the cancer cell itself. So are there risks? This is just a short story of a risk of a gentleman who had a reaction to his adenoviral vector for a disease he was getting some gene therapy from. So risks: the inserted DNA could activate your oncogenes or interfere with your tumor recessor genes. So this could increase your risk for cancer, because both of these genes, cause cancer to both of these cause cancer to occur in very different ways, and we talk about both of these in the week we talk about genetics and cancer. It can cause disruption of the gene's essential function, and this can cause some severe side effects. So with genetic testing, genetic testing can be done at any stage of human development. It's done for provides various views of the human genome. We can look at our ancestry, our health. We can see how we're similar, how we're different. We can look for mutations, for diseases, for many things. So why might a person choose to get some genetic testing? They have a family history of something going on, or they're just personally curious and they just want to know. So genetic counselor is a healthcare professional who will assist individual families with their genetic testing and results. They'll do a family history. They'll look to identify trends. This is that 3-generation pedigree that you'll hear about. That goes back 3 generations to look for disease trends. They'll look at that information and talk to the patient and then determine whether there should be some specific testing done. And they'll assist with after the testing is done, they'll assist with the interpretation and helping the patient interpret and understand the results, as well as the health risks and issues associated with the information. So there are types of genetic testing. I'm not going to cover every single type of genetic testing that's in the table, but I do encourage you to please be familiar with the different types that are on there. So we're going to talk about a couple. Preconception testing. This is where before a couple or parents decide that they want to conceive a child, they want to know if they are carriers of a specific disorder, such as Tay-Sachs disease, such as cystic fibrosis. Um, Both of those diseases are autosomal recessive diseases, so it would take two recessive genes to cause expression of the disease itself. They want to know if they're a carrier. And if we remember our Punnett square in genetics, so if both parents are carriers, with each pregnancy, they run a 25% risk of having a child that will have the disorder. They run a 50% chance that they will have a child that will be a carrier, and a 25% chance that they will have a child that have no, will be free of a carrier, will be a non-carrier. And we need to remember that in preconception testing, this applies only to each pregnancy. Each pregnancy carries the same type of risk. So let's say a family has both parents are carriers of cystic fibrosis. They could have four pregnancies, and all four pregnancies could get and develop cystic fibrosis. Just and then there's prenatal testing, which is when we look at fetal chromosomes for abnormalities. This has been going on since around the 1970s, well-known amniocentesis. We now can do chronic villi sampling. Both are invasive and have risks to mother and the fetus. Mothers sometimes get blood markers such as the alpha fetoprotein. However, if the markers are positive, they still need to go on and get the chromosomal testing, either with an amnio or chronic, and have the fetal cells analyzed. Um, there is newborn screening. This tests for errors in metabolism. PKU was the first to be identified. It will identify unusual metabolites or chemical imbalances. Please note that this is not DNA screening. This is only screening for errors in metabolism. It is not genetic screening. Each state has the ability chooses which newborn screenings it will do, and each state is different. So Washington doesn't screen for the exact same list that Idaho does. There's DTC testing, which is direct to consumer testing. This is for the average consumer. It does not fall under the regulations of clinical or laboratory amendments, which are run by the government. It also doesn't fall into any patent complications because it is for personal and educational purposes only. It's not to determine any type of DNA for medical research. 23andMe used to run a health analysis in their DCT testing. They are no longer allowed to at this point because many people were taking that and then not using it for educational purposes, but were trying to then treat themselves because they would get this, that they had a high predisposition to something. Um, There are nutrigenic testings which are related to diet, exercise, lifestyle, your nutritional. It looks at what types of foods or diets would be best for you based on your genetic analysis and what types of supplements. We also are using genetic testing to match patients to drugs, such as in a pharmacogenetic or pharmacogenomic test. This is helping to see if a patient would even metabolize a drug. Are they a rapid metabolizer? Are they a slow metabolizer? What is their, it gets into the P450 chromosome, P450 metabolism with medications. But we will talk more about that in our weeks with pharmacogenetics and pharmacogenomics. But just know that that research is out there and they're looking at that to help limit the trial and error of medication with patients. Um, There is a nice video in your content section of your week that talks about gene therapy and vectors and how they're used and how it gets the actual DNA in, and the gene therapy into the cell. So I would encourage you to watch it for knowledge. And I'll put a week 3 class question in the main discussion area of our class, and I'll check that frequently, or you're always welcome to email me if you have any questions. Thanks. Have a great